as the DNA replication continues, so as the helicase will actually unwinds the double helix DNA. It will continue to break the hydrogen bonds. And then single strand binding protein will bind to this region. And after that, okay, you can see that here you already have the free 3' ends. So there is no need for the primase to catalyze the RNA primer again. So that means the DNA polymerase tree will just continue adding the DNA nucleotides until the end of the DNA strand. Okay, so the elongation process, using this one as a template, so you can see that the replication continues, okay, towards the replication fork. This one will create a continuous strand which is known as the leading strand. But at this region here, you still need the primase to synthesize the RNA primer, since you have no free 3' end over here. So you still need the primase to catalyze the synthesis of RNA primer from this 5' end towards the 3' region. So primase will add the complementary RNA nucleotides, and after you have produced a 3' end over here, then the DNA polymerase can keeps on adding the DNA nucleotides. And as before, okay, it will not seal the fragments here because it will just add the DNA nucleotides, but there's still a space here. That means you will produce a fragments of DNA. When the replication occurs away from the replication fork, this one will create a discontinuous strand which is known as the lagging strand. So these fragments of DNA is known as the Okazaki fragments. So the hydrogen bonds is formed between the DNA. So now you have not still yet fully DNA double strand because you still have the RNA nucleotide over here, which must be replaced with DNA nucleotides. Okay, so the function of DNA polymerase tree is to add free DNA nucleotides to the short RNA primer by adding complementary DNA nucleotides to the bases of the template.